little bit of a mix-up, but thanks to the organizers, thanks to Bio-IT for having me. My name is Leon Spanner. I'm a data scientist at UCLA Health, but I also built EDA Toolkit with my co-founder of a new company that we're launching, Data Science Dynamics, Oscar Gil. And so you can use AI to accelerate, for example, vaccine design, things of that nature. What happens when the data that we are feeding these models isn't what we think it is or what we expect? So the green button, right? Oh, top green. So here's a headline number you're going to see in clinical ML papers right now. 93 to 94% accuracy. This is the band. You're going to find it in diabetes risk papers. You're going to find it on sepsis prediction papers, cancer screening papers, etc. Pick a journal, pick a month, you'll find it. And on the first read, that number does something to your brain. You see 94% and you think, okay, this is solved. We can deploy this. Can we? I mean, high accuracy is not the same as clinical signal. A model can hit 94% in a data set that has no underlying biological structure at all. If the labels and features are correlated by accident, then you might have a problem. Or if you have a synthetic data generation process, your model will learn that accidental correlation. And it will look brilliant on the test set. It will look brilliant in the paper. It'll just not help anybody when it hits a hospital. So most clinical ML papers skip data validation. That's the step that's really important in order to catch what I'm about to tell you. Because look, feature selection gets a section, right? Model architecture gets a section. Distributional validation, which is just asking, does this data actually contain the signal that we're expecting to predict, doesn't get a section. It gets skipped. And here's the real kicker. Bad data still hits the metric. And that reported accuracy that you get is real. And the problem is what you're asked to do with it is meaningless. So you publish 94%. And 3 years later, when someone tries to deploy that model in a real patient population, nobody can explain why. It falls apart. So trust starts before the model. It starts with the data. So the EDA Toolkit, validation before modeling. That's what we built. It's an open source toolkit that can exist to make the upstream validation step that you have easy enough that nobody has an excuse anymore. So for the next few minutes, what I want to do is focus on two specific checks from that toolkit. And in my experience, that's the difference between catching a problem in the data set and not catching it. So to make this concrete, the case study for this talk is a recent, from the last year, IEEE paper on pediatric type 1 diabetes. I'm going to keep coming back to it for reference. So first we have class separability. The toolkit plots the distribution of your features split by your outcome variable. So if you're predicting diabetes, you look at HbA1c in the diabetic group versus the non-diabetic group. If your feature can actually distinguish between those two groups, you should see two separated curves. You see one overlapping mass if it cannot. And there's no math you need to do. Your eyes do it. So second, we have cohort tables. the publication-ready Table 1 that every clinical paper in this room either has in it or should have in it. We made ours produce the significance testing automatically, formats the demographics cleanly, and surface patterns that should not be there. So once again, we have 2 checks, 1 toolkit, and we build this so that this step never gets skipped again. All right, so let me show you what happens when you actually point this at a published clinical ML paper. So in this paper, they had over 300,000 records, again, 94% accuracy. And the authors took this data set, they applied 4 different feature selection methods, they blended them into an ensemble, and they reported those numbers right there, 93.58 to 94.13 classification accuracy. On a type 1 pediatric diabetes data set. Their 2 top-ranked features were pancreatic function and BMI. Reasonable picks. But pancreatic function is obviously relevant to type 1 diabetes, and BMI is loosely associated with metabolic outcomes. Sounds plausible, perhaps, on paper. So we took the same data set, we ran the EDA Toolkit on it before doing any modeling at all, and here's what we found. So, 4 panels. You have HbA1c, BMI by diagnosis, age, and blood pressure. Each panel shows the probability density of that feature broken up by diagnostic class. Blue is no diabetes, red is diabetes. Look at the top left, HbA1c. That's the primary biomarker for type 1 diabetes. It's the thing that every endocrinologist in the world uses to make this diagnosis. If your data set has real type 1 diabetes signal in it, then the blue and red curves would be separated. They should look like 2 different mountains. However, they overlap almost perfectly. There's no class separability, especially on the primary biomarker for the disease that the model is supposed to predict. And it's not just HbA1c. The paper's top ranked features, pancreatic, BMI, they show the same identical distribution pattern. And if you were to examine the pairwise correlation between the clinical features, they approximate 0. So here's the verdict. The accuracy is real, but the signal's not. So these may be very well synthetic data artifacts, not clinical relationships. So if a clinician tried to deploy that model in a real pediatric cohort, the wheels come off immediately. And nobody publishing the paper would know until it happened. So the statistical tell, table 1. If the picture itself didn't convince you, the numbers should. It's the same data set, but different function. This is generate table 1, the output from the EDA Toolkit. It's run on the same north of 300,000 patients, and it's the table that should have been in the original paper, but it wasn't, and that's the whole problem. Look at the red p-values in that last column. The first thing I want you to look at is exactly that. They're all below 0.001 for BMI, HbA1c, and blood pressure. So if you're a clinical research, your instinct when you see that column is to feel good, right? They're statistically significant. Those are strong p-values. Those say that the diabetic group and the non-diabetic group are statistically different than those numbers. So the data must be real, right? No. So Welch's t-test will report a small p-value whenever two distributions have different means, regardless of where those distributions come from. So if you generate two synthetic distributions with slightly different parameters, the t-test will flag the difference as significant. The numerical output is real, but the clinical meaning is not. And that's the trap. Statistical significance and clinical signal are two different things, and the field unfortunately treats them as the same thing. Now look one row down, pancreatic. P-value is 0.36, not significant. That's the feature that the paper ranked number one. Number one. So according to our most predictive feature in the model, according to 4 different feature selection methods averaged together, and it has no statistical separation between 2 distinct groups, those 2 diagnostic groups. So the means are 0.97 in both, the standard deviations are 0.73 in both, and it's a statistical null result. You can't have a feature ranked number 1 in importance and simultaneously have it be statistically insignificant or indistinguishable between your 2 outcome classes. That just doesn't happen in real data. That happens in synthetic data. And you have identical group proportions. That's the third thing. That's the most telling one for me. Look at those proportions, 50.04 male percent in both diabetic and non-diabetic groups, 75.4% for children ages 5 to 11 in both. So the real pediatric type 1 diabetes doesn't behave like that. Type 1 diabetes incidence is strongly age dependent. It peaks in early adolescence, and so the diabetic and non-diabetic age compositions should look different. They are not, in this case, they are indistinguishable. So the synthetic data generator sampled both groups from the same underlying distribution, and which is exactly what gives you these matching proportions all the way down. That's the fingerprint. So once you see it, you can't unsee it. And what we want you to do is to be able to plug this into your pipeline. The EDA Toolkit is one source that works with any tabular clinical data set. And anyone on this list, pharma, biotech, clinical researchers, investors of the idea, that's who should be using it. That's how I want to bring it home. Because for pharma and biotech companies, this gives you the ability to validate the data set integrity before you even commit to a pipeline budget. You can catch corrupted data quicker, synthetic data quicker, noisy data, and so on and so forth. And of course, it's an invaluable tool for clinical researchers as well, because you can use this to get a quick table 1 result, which has been unit tested. It's been subjected to very rigorous coding standards, and so it's publishable. Here are the references. Number 1 is the IEEE paper I was just mentioning to you on the type 1 diabetes, and number 2 is our EDA Toolkit library. I want to say thank you to my colleague Oscar Gil for helping me build a phenomenal library, my colleagues at UCLA Health that gave me invaluable feedback, and of course the organizers for inviting me. Thank you.